slides today. So Joe, why don't you just go ahead and share your thoughts on bats and the role of environmental pathogen reservoirs in shaping the kind of disease outbreaks that we are having right now.
All right. Well, thanks again for inviting me to speak during your seminar. And I did just want to note if people have questions, and it's easy, you should feel free to kind of stop me as I go. I'm happy to clarify anything. So I'm sure we're kind of all aware now that emerging infectious diseases can pose a major threat to biodiversity. So just to kind of put it into context, thinking with kind of the current pandemic of COVID-19, mortality is somewhere between around 1% to 2%. However, for many wildlife diseases, which is really what I think about a lot and where a lot of my work is kind of focused, is on the wildlife end of things. Some of these diseases can cause mortality upwards of 90 to 95%, and in some cases even mortality. So we're talking about kind of regional population collapses and species extinctions in many cases. And I've listed just kind of a few major diseases here. These are kind of some of the major wildlife diseases that are causing huge declines globally. And so if a pathogen is actually able to survive in the environment, it can really exacerbate disease impacts. So we know that it can contribute to the potential risk of host extinction. And so it's really important in thinking about how do we actually control disease outbreaks. So this is a really classic example. This picture here is kind of referring to cholera, which is a globally important disease. And it can persist in the environment in this kind of water and be transmitted to people. And so a lot of the control practices are actually aimed at trying to clean up water and reduce the amount of pathogen present there. So I just want to go into kind of a little bit more detail again about exactly what I mean when I'm talking about environmental reservoirs and kind of transmission from the environment. And so I'm going to use kind of this just kind of bat example here. So basically, if we have a simply directly transmitted pathogen, that basically means when a host becomes infected, like we have here in this, I don't know if my arrow will work. Oh yeah, there we go. So we have this infected host here. And basically, for the duration of that host infection, it can basically transmit to a new host. And so basically, if that host recovers or if it dies, it's no longer able to transmit, which can give us leverage in trying to actually control diseases. However, if that infected or if that pathogen is actually able to survive in the environment, that a host can also transmit or shed pathogen into the environment where it can persist in the absence of that actual host. Where it can then kind of go on to transmit to additional hosts beyond the infectious lifespan of that kind of primary host that we were looking at here. So the system that I primarily work in is white nose syndrome. And this disease is characterized by a series of kind of seasonal outbreaks each winter, which is caused by a fungal pathogen called Pseudogymnoascus destructans. Now this pathogen, there's actually a picture on the right-hand side here that shows what one of these infected bats look like. And it really only affects bats, again, during the winter when they're hibernating. So this disease was actually first detected in 2006 in a single cave in upstate New York here in the US. And we now know that it was recently introduced to North America from somewhere in Eurasia. So again, the disease first emerged in upstate New York, which is indicated by this kind of red circle here, and then subsequently spread fairly quickly throughout kind of eastern North America, and is now spread through kind of most of most of the continent. So you can see each county here in the US is basically colored by the year at which the pathogen or disease was actually detected in that county. And so in its wake, this disease has caused really devastating declines to bats. So at least 3 species have declined to over 95%. And these are not just kind of rare species. These, prior to the arrival of this disease, were actually all growing, and we're actually doing really well, even with all the kind of adversity that humans are kind of putting on the environment and other species. And so one species has actually been predicted to go extinct from this disease, and so it is causing really severe declines. So we know that the pathogen is also distributed fairly widely across Europe and Asia. And to date, there's actually been no mass mortality. So we don't think that it's actually killing bats in the same degree that it is here in North America. And it's likely been present across that region for millennia, which recent genetic evidence shows. So I just wanted to kind of touch a little bit more on some of the background about the kind of seasonal patterns of this disease, which will be important kind of for the rest of the talk. So as I mentioned earlier, this pathogen can only infect bats during winter hibernation. And that's because the pathogen is basically psychrophilic, which means that it grows only at cool temperatures. And so when bats are active, their body temperature is somewhere between 37 to 41 C, which is really too warm for the fungus to grow. It can actually only grow below around 20 C. And so when bats begin to hibernate in winter, they actually cool their body temperatures down to about ambient, which is within the range of fungal growth. And so these infections kind of manifest over that winter period. If a bat is able to emerge in the spring, they can actually completely clear infections over the summer when they do warm their bodies up and that fungal pathogen is no longer able to grow anymore. However, when bats the following fall return to these hibernacula where they'll spend the winter and hibernate, they become reinfected from contact with that environmental reservoir. So we know that P. destructans can persist in the environment for really long periods of time, and that this is the kind of source for reinfection. So we know that it's important in kind of fueling that reinfection, but what the actual contribution of that environmental reservoir in actually driving the disease impacts is still unknown. So that's what I'm going to be talking about today. So this project, we basically conducted sampling of P. destructans in kind of a number of different ways, and I'll just kind of walk through what that is. This map here is actually just showing distribution of our sampling sites. So each one of these red circles with the yellow dots is a site where we sampled. So you can see we have samples across kind of eastern parts of the US, as well as a number of countries in Europe and Asia. And so we had 8 countries total where we sampled. We had roughly around 105 total sites. We sampled 47 different species across this range, which is about 23,000 total samples in the analyses that I'm going to present. So we collected swab samples from bats and cave environments, and this was done in early and late winter so that we could actually look at the change in later. We also simultaneously censused bat populations so that we could actually examine population impacts and link that back to the data that we are collecting from bats as well as the environment. All of the samples were tested using a qPCR, so this gives us not only whether the pathogen is present on those bats, but how much is actually there. And then all of the transmission and population data were analyzed using Bayesian hierarchical models. So I just kind of want to walk through what the change in population growth rates look like in North America. So I mentioned that there's really kind of devastating mortality, and so it's really important to kind of understand when that's actually happening. So this graph is basically just showing you different species down here on the x-axis, and each one of these panels is basically going to be kind of a different stage of the disease as it kind of moves through these populations. On the Y, we have population growth rate. And just to remind you, a population growth rate of 1 basically means that it's stable. So if these black dots are above the line, it means that populations are growing. If it's on the line, it means they're stable, and if it's below, it means that they've now begun to decline. So prior to the arrival of this disease, populations were either stable or kind of slightly growing. What's really interesting is that during the first year that the pathogen is actually invades into these populations, we don't really see any evidence of mortality. So all species are still basically about the same as they were prior to the invasion, and we don't really see substantial mortality. However, in year 2, so the second year after the pathogen invades into these populations, we begin to see major decline. So all species have now dropped down below that line. And then we basically see that consistent through years 3 and years 4. And so basically year 2 to 4 is when we're really seeing the kind of major mortality. And I've kind of made this little arrow at the bottom so that I can kind of bring that through throughout the talk to kind of help remind you when that mortality is happening. So what are the underlying transmission dynamics that are basically driving these disease impacts? Well, now I'm going to walk you through some of the transmission patterns, and this is really from the swab data that I talked about earlier. So I have kind of a series of graphs. I'll try to walk you through it. I know it's quite a bit here. So we have prevalence of the pathogen, which is the fraction of individuals that test positive for the pathogen on the y-axis here on the top. And then again, the same disease stages as I showed you earlier. And each one of these lines just represents the kind of model fit to each species, so we can look at those separate. And so again, prior to the invasion of the pathogen, there's no pathogen present in the population, which makes sense. When we see those stable populations, in the first year, we see that prevalence on bats is actually very low. But then increases to moderately high levels. So most bats are actually infected by the end of the first winter, despite not seeing mortality. And that is likely due to the long infection duration for this disease. So for P. destructans to kill its host, it takes somewhere between 90 to 120 days. And so that's actually a really long period of time, which means that most bats aren't becoming infected until this kind of period somewhere between January and February. And so if we look in the environment, we basically see a very similar pattern. So as you might expect, there's not really much pathogen present in the environment, but as emitting into that environment and really kind of ramping up that environmental reservoir. Now, if we look at what happens in year 2, we basically see a very different pattern. So now, in our winter period, and bats really start hibernating in November and kind of end in April. And so we see in year 2, at the start of hibernation, most bats are starting out already infected. And so we're seeing really rapid reinfection happening, which now means that bats are infected for a longer period of time, which is what's driving that really high mortality that we're seeing in that second year. And that's likely due to this kind of buildup of this environmental reservoir. So you can see the pathogen present in the environment just kind of continues to build. These different lines down here just indicate different types of sampling, but they're basically all showing us a very similar pattern. And so that is basically sustained across years 3 and 4. If we basically look at it statistically, there's actually not support for years 2, 3, or 4 really differing. And so we all kind of combine these later on. And it's important to note that, again, this kind of environmental reservoir just continues to build up, which is what's kind of driving that rapid reinfection that we see in bats. So what about populations and transmission dynamics look like across Eurasia? Well, if we look at population growth rates for species across Europe and Asia, we actually see that they're fairly stable. So again, we know that the pathogen is present in these populations. And although there is quite a bit of variation, which I'll try to explain a little bit later, if we kind of look on kind of a species level, we basically see in general, most species are fairly stable across this region. So now I'll kind of walk through transmission dynamics for just one country. And so here I'm looking at, this is actually data coming from China. And so what we see is that it actually looks very similar to that first year in North America. So I'll just kind of bring that back in so we can see most bats are not infected in early winter, but then we see by the end of winter, they kind of slowly ramp up, which means that bats across this region are really not infected long enough to actually result in mortality, similar to what we saw in North America. And now just to remind you, it's not that bat species that these sites are just being invaded by this pathogen. We know that it's been present here for a long period of time. We actually conducted our study over multiple years. So here's 3 years worth of data. And you can see that same kind of cyclical pattern that's happening year after year, where bats start out with kind of low amounts of infection in early winter, and then it kind of increases. Now, I've included the substrate data on this graph just for ease of looking at fewer graphs. And you can see that what's really interesting is that the environment actually starts out really low. And one kind of really important thing to note is that over the summer, we're actually seeing a drop in the amount of pathogen present in the environment. And that's basically happening each year. And so what that means is that this lower amount of pathogen present in early winter is basically decreasing the probability that a bat will actually bump into one of these contaminated locations. So I'm just going to kind of bring all the data together now. So here's the data that we looked at from the US, as well as data from species in China. And bring in species from other regions, we basically see a really consistent pattern. So basically across the region where this disease is endemic, we're basically seeing bats having this kind of slow and delayed infection, which means that there's never really enough time for mortality to really occur from these infections. And then if we take that one step further, we can look at the relationship between the extent of the environmental reservoir, or the intensity, and the actual population impacts or change in population growth rates that we're seeing across all of these regions. And what we see is there's a significant negative relationship here. And so basically what that's saying is that as the environment becomes more contaminated, populations have lower population growth rates. And what's interesting is that this pattern was actually consistent across North America as well as Europe and Asia, which might suggest that as a site becomes more and more contaminated, even in the endemic region, it may still cause some mortality to populations there. So again, just to note, the key difference between these two regions was that over the summer, we saw the amount of pathogen present in the US basically stable or slightly increasing year after year, whereas across Europe and Asia, it's basically declining, which is decreasing the probability. Just to kind of summarize that, we saw a reduction of P. destructans in the environment each summer, which delayed infection in bats over the winter and reduced mortality. And that's really due to this long delay between infection and mortality for this disease. However, in North America, we saw this kind of continual buildup of the pathogen in the environment over years, which resulted in really early reinfection and very high mortality. So now I'm going to shift gears to another project, which is really kind of looking at the role of both direct and indirect transmission in driving disease outbreaks. So similar to what I just kind of talked about, but using some kind of more experimental approaches, and we're going to explore some of that data in a little more detail. So when trying to understand a disease system and how we might try to control disease outbreaks, it's really important to understand what host interactions will actually lead to pathogen transmission. So as I kind of already mentioned a little bit before, there's kind of two major modes. There are some other ones, but I'm just going to kind of focus on the most common ones, which is direct transmission. So that's just physical contact between two individuals that actually leads to pathogen transmission, and then indirect transmission. And this is basically pathogen transmission that's being mediated through some environment, like a door handle or some other surface. So Most studies that are actually trying to estimate what transmission might look like in a population typically just focused on direct transmission. And so we use things like contact networks as proxies for what transmission will likely occur and really estimate how much spread will happen. So there's some limitations to that approach. So obviously, as I mentioned already, they're missing indirect connections, right? So anything mediated through the environment is being completely missed. And we're also missing things like infrequent connections, and these tend to be kind of rare movements and interactions that might not happen that often, but can be really important in kind of fueling or sustaining outbreaks. They're typically things like interspecies interactions, like spillover, which helps kind of bridge gaps between populations and species and can sustain a disease outbreak that way. So collectively, I'm just going to refer to these as cryptic connections. They're things that we typically don't measure when we're trying to actually understand how a pathogen will spread through a population. So how important are these cryptic connections, and particularly in relation to the transmission of P. destructans? Well, we know already that some highly impacted solitary species, some solitary species are really highly impacted, which raises really important questions of how they're actually acquiring infection. So the approach that we're going to take is we actually measured connections using a surrogate pathogen, and this includes both direct as well as those cryptic connections. We also then measured connections using observational data, and this was done two ways. And it's important to note that this just includes those direct connections that are typically characterized to actually understand how a pathogen will spread through the population. And it's important to note that these are pretty typical of how we might try to estimate, say, with the current pandemic, something like SARS-CoV-2, how it would actually spread through a population. So this is something that's done for human diseases, agricultural diseases, as well as wildlife. So then at the end of this, we're going to basically compare all of these data to the actual disease epidemics that occurred in these same populations, so that we can basically understand what basically more accurately is actually characterizing the real disease outbreaks. So a little bit more detailed methods. So the study was conducted over the winter period, again, when bats are naturally infected with this disease. We did this study in Wisconsin and Michigan. Each site had about 3 to 4 species, so ranging from 20 to about 700 individuals. We traced connections for 3 highly impacted species, which range in their kind of social behavior, and I'll talk about that a little bit more. And then the surrogate pathogen and contact tracing were all done prior to the invasion of the pathogen. So first, I'm just going to look at connections using the surrogate pathogen. So we used an ultraviolet fluorescent dust as our surrogate pathogen, mainly because it really mimics the transmission pathways of PD. So P. destructans is an epidermal skin pathogen, and so it can be transmitted by direct contact as well as through the environment, as we already discussed. And so this dust makes a really good proxy for that. It also comes in different colors, so we can actually track individuals. So we actually marked 7 bats per site, each with a unique color. And you can see that process happening down here. We basically kind of evenly coat a bat of a particular species in one of these colors, and then release them back into these hibernacula and allow them to kind of go about their normal activities over the course of the winter. So again, we dusted bats at the beginning of hibernation in November. Then we returned and surveyed at the end of hibernation. You can basically see that dusting process happening here again. And then this is what a cluster of bats actually looks like at the end of hibernation when we come back. So you can see this yellow bat got in here and really spread its dust around. So again, we recorded all the colors of dust found on bats, and that's how we're actually estimating these connections among individuals. And we also looked at this in the environment as well, so we could characterize that. It's important to note, I'll mention it later on, but we measured all environmental contacts, which were also spatially mapped. So now I'm going to look at connections using our kind of observational data, or the kind of classic way where we're just looking at these kind of direct connections. And so I just want to kind of start with the workflow. And so this is what bats kind of typically look like in winter. So you can see we have this kind of large cluster here. There's kind of a single individual here. And so we can basically go in and kind of characterize all of these connections among individuals and basically convert that into these contact networks. So this is just a graphical representation of what one of those hibernating contact networks look like. And so in this, each one of these nodes is basically a bat, and the color indicates the species. One of the edges between two of these nodes indicates a physical contact, and all of the bats that are basically connected in one of these groups indicates kind of a cluster, as I showed you earlier in that photo. So we actually characterized these contact patterns in two ways. And again, these are kind of ways that are typically done when trying to understand transmission in human diseases. And so the first one is through direct physical contact. So just looking at contact tracing, which is, again, I'm sure a term we're all really familiar with. So that just basically means this highlighted individual here is in physical contact with just two individuals. Now, we also estimated shared group connections, which is basically saying this individual is only touching two, but it's actually in this shared group with, say 12 other individuals. And so it's likely that they're all sharing pathogens. So it's a little more inclusive measure. And so it's important to note that bats basically spend 99.5% of their time in these networks over the winter, but we do know that bats do periodically arouse, which would allow for them to make additional connections. So we really wanted to account for that. So we basically conducted a simulation on these networks as they would over the course of the winter, which is when we're really collecting that dust data. So we basically conducted this simulation and tried to guide it with as much biology as possible. So we collected together, looked at changes in their clustering behavior, so are there more or less clusters, and then how many arousals we needed to account for. So basically, the product of that is what I'm showing you here. This is basically one of these simulated networks, and this is basically just for 7 individuals within that network, but it's basically showing all the additional connections that they made through direct physical contact or shared groups over the course of that winter. Now we can compare those to our surrogate pathogen connections, and as you might be noticing already, there's far more connections estimated using the surrogate pathogen. Now, I mentioned our site and so we wanted to be able to compare across species and different sites. And so we basically condensed those network data down into what I'm showing here on the y-axis, which is the fraction of each species that's connected to a single individual, whole in a bunch of the other graphs. So basically down here, this is showing the focal species. So this is Mitis sucificus. And the color of the box above it means the species that it's contacting, or the dust was resited on. So this means that Mitis sucificus transmitted dust to other Mitis sucificus. And this point here would indicate that it transmitted dust to about 50% of the other Mitis sucificus at that population. And so again, this is with move over one to the right. This is basically now among species transmission. So this is dust that's being resited on a different species, Mitis septentrionalis. So now we can kind of bring that all together with our other measures of connections. And again, what you're hopefully noticing already is that the UVF-dust basically estimated far more connections, both within as well as among species. Really just overestimate the amount of connections, that there's actually proportional differences. So for instance, we see a really large amount of connections happening with the Obtuscus fuscus estimated through the dust. So in some cases, is almost 75% of that population, whereas we never really see that in either the shared group or physical contact. And then the same goes for things like Mitis septentrionalis. So we're really seeing a lot of transmission happening both within and among species once we start to include those cryptic connections. So what do the connections look like for other highly impacted species? So for Mitis septentrionalis, again, this is actually a solitary species. What we see that's really interesting is that it's actually very specific. So we see a really large amount of transmission. Again, the red to red means within species. We see a large amount of within species transmission. On average, about 25% of the individuals are connected to just a single individual bat in that population. And what's interesting about this as well is that we also see a fair amount of transmission happening to other species, in fact, a little bit higher than within its own species, which could explain some of the high mortality that we're actually seeing in this. Now, if we look at the last species, we actually see a very different pattern. So basically, our different measures actually didn't really give very different results. So whether we're looking at connections with the dust or through shared group or physical contact, we basically don't see anything that's really that different. And so this basically means that cryptic connections were not really that important for this species. But it also raises really important questions about what's driving the reduced connections in that species, Parametis subflavus. So I mentioned earlier that we also characterized the dust in the environment. And so we basically looked at whether environmental contamination could actually be driving that pattern. So we know that the environment is really important. And so is it possible that that species is just depositing less dust in the environment, which means it's being spread around less? However, we basically didn't really see any large significant differences. You can see Parametis subflavus is trending a little lower, but there's certainly individuals that had a fair amount of dust, almost the same as septentrionalis. And so that alone cannot explain these kind of broad scale differences that we're seeing in connections. So it's possible that the location of the contamination could be important. So again, I mentioned we spatially mapped each one of these contexts. So we basically estimated the area of space use for each individual, and then looked at the area of spatial overlap between those individuals to get a sense of whether Parametis subflavus, this species that had reduced connections, could just be occupying a different space from other individuals, and that's reducing them. And that's basically exactly what we found. You can see these two species have really high areas of spatial overlap, whereas Parametis subflavus has kind of greatly reduced overlap. So this basically means that spatial segregation can reduce the effects of these cryptic connections, as well as direct connections as well. So how do these data compare to the invasion of a real pathogen? Well, basically, we can look at the relationship again between P destructans and these various metrics that we calculated earlier. So this is basically on the x-axis here, I have the change in shared group connections, which I'm just going to show you the shared group versus the physical contact because that one actually performed a little bit better. And then compare that to the actual change in pathogen prevalence. So this is actually, we basically monitored these populations for several years after this study until the disease actually invaded. And then we were actually able to calculate the change in prevalence from early to late winter and then compare that to our shared group connections. And what you can see is that it's actually not really a good fit. So it's only explaining about 20% of the variation that we're actually seeing in the real disease outbreaks in these populations. So that means if we had simply used shared group connections or these direct physical contact metrics, we would have drastically underestimated the outbreak in these populations. Now, if we look at the UVF-dust connections, we see a very different pattern. So I've actually plotted a one-to-one line here. And you can explain much more of the variation, about two-thirds more variation can be explained by the UVF dust connections than by shared group, which means that we're not simply just overestimating the amount of connections, we're actually likely cap them. So just to kind of summarize that, so we looked at we found white nose syndrome epidemics cannot be fully explained without the inclusion of those infrequent and indirect connections. So basically including a lot of that environmental transmission helped explain how an outbreak would actually occur in that population. We saw cryptic connections were not simply proportional to the observed connections, and the importance of cryptic connections differed among species, and we saw that all types of transmission were reduced by basically spatial segregation. So I just wanted to kind of briefly mention, given everything that's going on now, some other projects, things that myself and collaborators have been talking about, is looking at both kind of somewhere along the lines of pathogen adaptation and transmission. And so for SARS-CoV-2, we know that it likely originated in bats and then was either transmitted to humans or transmitted to some other intermediary animal host, which then spilled over into humans from there. And so some really important outstanding questions, both from a conservation standpoint, as well as from being able to kind of understand and kind of control this pandemic, is one, can bats actually get infected with SARS-CoV-2 in its current form, so which may render it unable to actually infect its original. Can they actually transmit? So there's actually quite a bit of conservation concern about whether this virus will actually spill over into wildlife. And so this is a really important from understanding the spillover potential and kind of ecology of the disease, how well it's actually able to infect some of the original hosts that it may have originated in. And so with that, I just want to acknowledge all of my collaborators who we've worked with both here in the US as well as across a number of countries in Europe and Asia. And then my funding, most of my work is funded through the NSF Ecology and Evolution of Infectious Disease Program. And with that, I'll take any questions anyone has.